So our first speaker for this session is Marcelo Barria. He is from the National CJD Research and Surveillance Unit in Edinburgh, UK. He has a very important talk, public health-wise, entitled Assessing the Replication Properties of Prion Protein Aggregates Found in Archized Formalin-Fixed Paraffin-Embedded Appendix Tissue Collected from Asymptomatic Population Using Highly Sensitive In Vitro Prion Amplification Assay. Thank you.
Well, thank you for the introduction, Dr. Appleby. I know the title is quite long, and thank you for choosing this session to follow my presentation. First, well, I want to say that I am very honored and happy to be presenting our work today. So thank you very much for the invitation. And to start my presentation, I want to use the same slide that you probably saw at the resume part a few minutes ago. So I just did some modification just to resume a little bit and introduce the topic that I will be talking for the next 15 minutes. So the important part here is discuss about variant CJD. And variant Creutzfeldt-Jakob disease is a human form of a prion protein linked with the consume of meat products contaminated with an animal disease in cattle called bovine spongiform encephalopathy, or BSE. Despite the widespread of the exposition for the UK population because the BSE, today 178 cases of variant CJD have been reported in the UK. And in an attempt to understand a little bit more about the magnitude of the exposition of BSE in the population, 3 large studies, retrospective studies, were run to evaluate the presence of the abnormal form of the prion protein in formalin-fixed paraffin-embedded appendix tissue collected from asymptomatic individuals. So the final aim of this analysis was to evaluate the potential prevalence of subclinical infection with prions after the exposition with BSE. So the first question here is why we were, or these studies were investigating the appendix. Because, as we know, in the majority of the human prion diseases, the replication of prions occur in the central nervous system, and particularly in the brain. So, but one of the characteristics related with variant CJD, talking about the disease pathology, is that for variant CJD, after the exposition of the infectious agent, the prions replicate in the peripheral tissue, and one of them is the actual appendix, and then travel to the brain and produce some replication and damage there. So, taking appendix tissue, it's a valid way in how we can evaluate the position of the abnormal form of the prion protein. So we will concentrate more in this appendix 2 study because the samples that we use for my project was related with samples derived from this study. So now, more specifically, this study, the appendix 2, was analyzing anonymized formalin-fixed paraffin-embedded appendix tissue from around 30,000 individuals. And how they assess the presence of the abnormal form of the prion protein was using histopathology. So they were taking really thin sections of the appendix tissue and then evaluating for the presence of the abnormal form of the prion protein in, under the microscope. And this is just one example how an appendix tissue looked like under the microscope. What the scientists were looking for is these aggregates, and that is the abnormal form of the prion protein, so this sample was considered positive. So what the study concluded, so from the total of around 30,000 individuals analyzed, 16 were positive for abnormal prion protein. Indicating an overall prevalence of 193 per million population. In more simple words, 1 in 2,000 people in United Kingdom were having the abnormal form of the prion protein in the appendix. So Now we need to discuss a little bit susceptibility. I mean, what are the factors, the many factors involved in the susceptibility of variant CJD, for example? And one of them, as Richard mentioned this morning, is related with genes, particularly with the prion protein gene. So, and more specifically about codon 129, because I mean, one little segment of this prion protein gene corresponds to a codon, and the codon that is linked with the susceptibility is the codon 129. So this codon can be expressed in the population in different forms. So there are some individuals that are methionine at the position of at the codon 129, where there are other individuals, they are valine at the position at the same codon. Other individuals have, they are heterozygous for this polymorphism. We call that polymorphism. And they are methionine valine, so they have the combination of the two. Now that we know that the population can be distributed across these different polymorphisms, then we can link this information with variant CJD. So in variant CJD, today we know that 177 of the cases confirmed for variant CJD are methionine at the position of the codon 129. Only there is just one case which is pathologically confirmed, having the heterozygous polymorphism at the codon 129, and there is no cases of variant CJD linked to valine polymorphism. So now if we go back again to the Appendix 2 study, and now we dissect a little bit and try to consider what was the distribution of the codon 129 for the 16 appendices that were positive for the abnormal form of the prion protein, we can say that 8 of these individuals were MM, 4 were MB, and 4 were BB. As I mentioned previously, there is no cases of variant CJD linked to this codon. So what that means, what it means that this abnormal form of the prion protein was present in these individuals, and that still is an open question. And it's part of the project because, I mean, the samples that we used to analyze in this particular project was to evaluate the molecular activity in vitro of these 4 samples. So problems, usually scientists are, facing problems, and this was not the exception. And we have the first problem here. And the problem was related with the amount of tissue available to run these experiments. So the amount of tissue available today to run this experiment is extremely low. And what I show you here is a block of tissue that usually is cut, and these small and really thin sections are the ones that are evaluating under the microscope. So we receive a single curve of tissue to evaluate the molecular activity of these samples. So we didn't have too many chances for an error here. It was just one sample per individual. So in order to have a very good system, an optimized system that we are not going to lose the sample in case we fail. So we try different protocols first in how to extract the abnormal form of the prion protein from this tissue. We try methods that use alcohols, but finally the more efficient method was incubating this material just using hot water. And incubating that several times and centrifuging this material and sonicated, we optimize a protocol that was efficient to recover the abnormal form of the prion protein. The second problem, it was not an easy project, I can tell you. So the second problem was the amount of abnormal PRP in the appendix tissue is extremely low. So after the extraction method, we need another way to propagate this material in order to be detected. Conventional biochemical assay were unable because, have the limitation in the sensitivity to run this. So we decided to use a method called protein misfolding cyclic amplification. And we optimized one of these methods in the lab for high sensitivity, and it's called highly sensitive protein misfolding cyclic amplification. So what I want to tell you about the method, I am not going to talk details about the method, but in simple words, this method have the possibility to incubate the misfolded form of the prion protein and put it in contact with large quantities of the normal form. That's the only thing that we need to know. And after the several cycles of replication and incubation, the abnormal form is able to convert the normal form, and we are able to detect it. As simple as that. So after the amplification process, we run a technique called Western blot. And what do you have to take in mind for the following slide is that when we prepare this experiment, we take a sample previous to the amplification and one sample after the amplification. So we visually can evaluate if there is an increase of the signal. So for the following slide, if you see that there is a strong signal on the PMCA sample, means that the sample was positive, so propagate. So black, positive. So we try, after we optimize the structure method, we use brain from a Barria CJD patient, actually from four different patients. And we extracted the abnormal form of the prion protein from these 4 individuals. We also evaluate 2 negative controls here, and you can clearly see here now that the strong signal suggests that all the samples show the position of the abnormal form of the prion protein after the extraction and after the propagation. So our system was working pretty well. So the next step was to evaluate now appendix tissue that is more related with the actual project. So we ran a similar approach. So we selected 4 cases, and after several cycles of amplification there, we were comparing the frozen and the sonicate samples, and the sonicate samples show that are able to propagate prions in the 4 cases that we selected. So the system was able to propagate not just brain material, but also appendix material from Barria CJD cases. The negative controls didn't show any propagation, as you can see there. So at that stage, we thought, well, we are in a really good stage because we have a very efficient system that is able to extract the material, and we are able to propagate that. But again, there is another problem. And as I mentioned previously, there is no Barria CJD cases related with BB. So we didn't have really a positive control to run our experiment. So what we decided to do was quite ambitious, but from a very simplistic way. From a molecular perspective, we were taking now BSE and exposing the 4 different proteins there to evaluate the susceptibility of this protein to be converted by BSE. You will understand now why we did that. So we use PMCA to do that, so incubate the normal form with BSE and see what happened. As we expected, the PMCA conversion showed that the codon 129 samples were efficient in propagating prions. As you can see here, it's more black here compared to that and less there. So the red correspond to the codon 129. MB was a little bit less efficient, but the BB codon was, very poor amplifying the abnormal form of the prion protein derived from the BSE. And this is the material that we were interested, but potentially this could mimic the material that we may found in the appendix tissue of the BB individuals. So after to have that material, we decided to now evaluate what is the molecular properties in terms of replication of the material that we created in vitro. And again, we incubate this abnormal form of the PRP with codon BB, now with the 3 different polymorphisms, just to see how compatible are this material with the 3 different forms of the protein. And what we found was this. I mean, the material that is carrying the codon 129 BB was able to propagate in all, but now it's the BB protein that is more susceptible for conversion. So now putting all the pieces together, we were now at the time in where we can extract our material and then propagate with the best substrate to see if we can able to amplify the appendix tissue derived from the appendix 2. So this is the final experiment. So we ran the experiment, we extracted these 4 samples that were codon 129 BB. We ran a PMCA. We use controls, potential controls here. So we use appendix brain, appendix and brain from a Barria CJD case, but with the caveat that these Barria CJD cases are methionine for the codon 129. But in technical terms, it's a good control. Then also we decided to include some sporadic CJD and a non-CJD brain as a negative controls. And after a single round of PMCA, we extracted the material, we ran the amplification, and any of the cases show any propagation in vitro. In order to increase the sensitivity, we ran a second round of PMCA. And as you can see now, the controls are working stronger here. So, I mean, they work really well, the controls, where the experimental samples, again, didn't show any propagation. And finally, we decided to run a third round of PMCA just to see if increasing the sensitivity of the amplification, we were able to see something. And this was quite consistent with the second round. So any of the samples that were derived from the appendix 2 samples carrying the BB codon 129 amplify in vitro. So there is a lot of discussion need to be made here to understand what are the limitations of the technique and the potential conclusions for this. But one of the things that we believe today is that the distribution of the abnormal form of the prion protein in the appendices is not homogeneous. So there are high chances that we are taking just one little piece of tissue that doesn't have actually the abnormal form of the prion protein. So that probably is one of the reasons why we're not able to propagate in vitro, but it's something that we are testing currently. So, summary. So we developed a robust protocol to retrieve abnormal prion protein aggregates from archived formalin-fixed paraffin-embedded tissue section from Barria CJD brain and appendix tissue. We now have a platform for the analysis of the residual formalin-fixed paraffin-embedded tissue specimens from the appendix 2 and appendix 3 studies, and these analyses are ongoing currently. And related particularly with this project, so the HSPMCA analysis, so the amplification analysis of these 4 cases, codon 129 BB, anonymized appendix tissue derived from the appendix 2 studies do not show any in vitro amplification activities after 3 consecutive rounds of amplification. Interpretation of these results, as I mentioned previously, are ongoing and may require additional analysis. So with this last slide, obviously I want to thank you all, but especially the people that contribute to fund these studies. And also I want to thank people that actually is linked with this study at the National CJD Research and Surveillance Unit, and particularly the people that actually run the experiment. So is Dr. Suzanne Suleiman, Fraser Bryden, and with the contribution of Dr. Diane Ritchie. And with this, I just want to thank your attention.